Some of you may have heard of the name Louise Brown. She was the first test tube baby, or in vitro fertilization baby, and she was born right here in the UK. What some of you might not know is that in vitro fertilization, or IVF, isn't only for couples who are infertile. But why would a fertile couple want to go through the complicated and expensive procedure of IVF? Well, the answer to that is hereditary genetic disease. A couple with a genetic disease is able to use IVF to produce a genetically healthy child. And this is done through a process called embryo biopsy. You take a sperm from the man, an egg from the woman, and you fertilize them together in the lab to produce an embryo. This embryo divides into 2 cells, 4 cells, 8 cells, and so on. And at this early stage, every single cell in the embryo is perfectly identical. And what we know as scientists is that at this stage, you can take one of these cells out of the embryo, and the rest will continue to grow unhealthily. And with this one cell, you can test the entire genome of that whole embryo. That way, you can choose an embryo that's free of the genetic disease and place that one back into the mother's uterus. So how do you get the cell out for testing? It's a really delicate and fragile procedure because embryos at this stage have a protective shell around them, much like the shell of a chicken's egg, and this needs to be breached. Currently in clinics, you can do this by drilling a small hole with a low-level laser, and that's where my research comes in. I'm researching the safety of these lasers on embryos. Embryo biopsy is performed all over the world, and healthy babies have been born. But how safe is this technique really? There's no standardization or detailed safety investigations of how that hole should be created, and every clinic does it differently. So to investigate the safety of these lasers on embryos, I'm using a mouse model. It looks and grows the exact same way as a human embryo in these first days. I'm testing different laser hole sizes, laser powers, and hole locations in this shell to see if there are any damaging effects that we don't know about yet. Results so far at the halfway point of my thesis have found there are some laser parameters which appear safe for use in embryos, and others which appear damaging in terms of the quality of the DNA and the embryo's metabolism. I'm currently looking to see if there are changes in important developmental genes that are having negative effects. My goal for the future is to find safe laser parameters that can be standardized for the clinic, and to highlight other damaging parameters that should be avoided altogether. Given these procedures are used to literally create healthy life, the safety of each step is of the utmost importance. And with the continuation of studies such as these, we can have the peace of mind of knowing that these laser techniques are not damaging the lives that are being created. Thank you.